All right, everybody, welcome back. So, we're going to pick up where we left off and talk about how we are going to produce the amazing energy molecule known as ATP. So, we talked about the fact that you are going to produce the ATP via cellular respiration in order for the ATP to be used during cellular work. Okay, so we're going to talk about the ways that organisms will produce ATP, and there are three main ways. First, the substrate level phosphorylation. Second is oxidative phosphorylation, and third is photophosphorylation. And basically, the difference is between all three is the source of the energy that is used to create the ATP molecules. So we're going to be talking about those individually. So we're going to start off with substrate level phosphorylation. Now I know that sounds like a lot and everybody's like freaking out right now. Wait, what does that mean? It's very, very simple. Okay, if we break down what these words mean, substrate, aka the reactant. So at the level of the substrate, you're going to phosphorylate a molecule. That means you are going to take a phosphate from the substrate or the reactant to produce the ATP molecule. So what happens is you are going to break a bond, okay, of a molecule that has a phosphate on it. So you're going to break the bond between the molecule and the phosphate that is attached to it. So let's say you have this substrate. Let's say it is a glucose 6 phosphate. Let's say it's glucose with a phosphate on carbon 6. Okay, glucose 6 phosphate is going to be broken. So you're doing a hydrolysis reaction. You're going to release the energy and you're going to release the phosphate group. You're going to break the phosphate group off of the glucose molecule. So then you have glucose. Okay, so you have the glucose molecule, and then you have the ADP, which is found hanging around, which we'll talk about later on. And then you take that phosphate from your original substrate and you add it to the ATP, the ADP molecule, excuse me, to produce ATP. So the phosphate is being released at the site of the reaction. Okay, so basically, substrate level phosphorylation. Again, you're removing a phosphate group from a substrate and you're adding it to ADP, which is adenosine diphosphate. There's only two phosphates on it. Then when you add the third phosphate right here, then you produce the ATP. Okay, so oxidative phosphorylation, okay, is the second type. And we're going to be talking about this in great detail when we get to the electron transport chain. And basically what happens is you are utilizing redox reaction. So remember, reduction, you're going to be gaining an electron. Oxidation is where you are losing an electron. So a molecule is oxidized, okay? So you're losing electrons. The electrons are being released from the bond, okay? And then you are going to be reducing a molecule. So something's going to pick up those electrons, and they are usually the electron carriers, the NAD plus, the FAD plus, the NADP plus, which we're going to be talking about, okay? And what you're doing is the electrons that are being generated and released, you're also going to be releasing protons with it, and the protons are going to move via a proton motive force because you're going to create a gradient, and the protons are going to want to cross the membrane. But they cannot cross the membrane because remember, protons are ions and ions are big and they are polar. and we have that barrier in the membrane. It is the lipid part of the phospholipid bilayer, okay? So remember the redox reactions, okay? So redox reactions, oil rig, so oxidation is losing. So what are you losing? Electrons. Rig, reduction is gaining. Or another one that I learned in high school is Leo the Lion says grr, okay? So losing an electron is oxidation, gaining an electron is reduction. So this is what we're talking about when we're talking about oxidation and reduction. Oxidation has nothing to do with oxygen. Reduction has nothing to do with the size. It simply has to do with the addition or the loss of electrons to a molecule. That's what it is, okay? So, for example, in redox reactions, okay, we have the process of cellular respiration. It's basically a series of multiple redox reactions. When the bond is broken, the electrons are released. So again, you're losing the electrons. So that's oxidation. You have the electron carriers, NAD, FAD, NADP, picking up the electrons. And when they are picked up, okay, they are reduced. So the electrons were picked up by NAD plus, and they become NADH, which is reduced. And what's going to occur is those electron carriers, they're carrying electrons, they're going to transport those electrons to the electron transport chain. So when NAD receives the electron, okay, it has the hydrogen, because we're going to talk about later on, electrons are not on their own. We do not have the ability to undergo subatomic reactions. This is all basically the movement and transfer of hydrogens, okay? So then you have the production of NADH, which is then going to carry that electron to the electron transport chain. And we're going to talk about that in great detail. So you can look at another diagram basically showing you the reduction of the NAD molecule, the NAD plus, which is the electron carrier. So to go from NAD, okay, to NADH, you are reducing it. So the electrons are moving to NADH. And the opposite direction, if you're taking NADH and it's giving the electrons away, it's going to be oxidized and you have the NAD plus again. We're going to talk about how that occurs during the electron transport chain. Don't freak out about this diagram right now, but what I do want you to know is what was said here. Reduction is where you're gaining an electron. So now you're adding the electron here, and basically you get the new molecule, NADH, it's carrying the electron, and then you can oxidize the molecule because you're going to give up the electron and it comes back to NAD plus. So the proton motive force that I discussed earlier, also known as PMF, is basically this. You're going to produce a gradient of hydrogen ions on the internal side of the membrane, on one side of the membrane. Okay, so let's say this is internal and this is external. Okay, so you're creating this gradient. Now, from what we talked about transport across membranes, what do you want to happen when you have a high number of molecules on one side of the of the membrane? Exactly. You want the molecules to cross the membrane due to the law of diffusion. You want to go from an area of high concentration to low concentration until equilibrium is reached. Now, hydrogen, or protons, because they are ions and they are positively charged, that means they can interact with water, they are polar. Therefore, it cannot just cross via simple diffusion. We need to facilitate it. So it's facilitated diffusion. So you have a protein, which is a transmembrane or integral protein right here, known as ATP synthase. So the one hydrogen that crosses over is going to power taking the ADP and the phosphate, bringing them together to form ATP, adenosine triphosphate. Okay, so we're going to talk about that when we get to electron transport chain. So just to let you know, for your information, one hydrogen ion can fuel the production of one ATP molecule. It's basically what I showed you here. One crosses, you get one ATP. Another crosses, you get another ATP. Another. So if you have 8 hydrogen ions, 1, 2, 3, 4, 5, 6, 7, 8 here, how many ATP are you going to produce on the other side? Exactly. You're going to produce or generate or synthesize 8 ATP molecule. Okay? So with oxidative phosphorylation, again, continuing, it's basically the movement of the protons, which are going to generate the ATP molecule. Last, photophosphorylation. So let's look at that. Photo, so light, phosphorylation, the phosphate. So you're using light to add a phosphate molecule to another molecule, which in this case is we're going to produce ADP. So the electrons are going to be energized via the light from the sun. So we're going to be talking about photosynthesis in great detail. So if it's photophosphorylation, it's photosynthesis. Yes. You can see Mr. Sun right here. Okay. So we're going to talk about what all this means, but we just want to go over the definition of photophosphorylation right now. I'm going to talk about photosynthesis again in the next chapter. The electrons are energized via the light from the sun. The electrons will then generate the proton motive force, which will then allow the production of ATP. And that is what photophosphorylation is. Again, we're going to talk about it when we get to chapter 7. So again, to review the 3 ways to make ATP. 1, substrate level phosphorylation. You're taking the phosphate off of the substrate to produce ATP. So that phosphate is going to come off, it's going to be added to ADP, and then you're going to produce ATP. Second way, oxidative phosphorylation. The electrons are going to be generated via the oxidation, and basically you're going to create a proton motive force, which is going to want to cross the membrane, the protons. So the proton motive force basically is going to power the ATP synthase molecule that is embedded in the membrane and cause the phosphate and the ADP molecule to come together to produce ATP. Finally, the third way to produce ATP is the photophosphorylation, which is using the sunlight energy plus the proton motive force to take the ADP and the phosphate to bring them together to produce ATP. So again, remember, the difference between all 3 of these is the source of energy used. Please remember that. Okay, so now I want to get into the discussion of how we are going to generate or produce or synthesize this energy. And basically the way that we do that is through two main processes: photosynthesis and cellular respiration. Okay, with photosynthesis, you're taking the light energy, so you're taking the energy from the sun, and you're producing chemical energy. The overall process, now we talk about overall, on a whole, the overall process is endergonic. That means energy has to be put into the reaction in order for it to occur. Cellular respiration, on the other hand, is an exergonic reaction. And what you're doing is you're releasing the energy from the molecules, okay? And basically, again, you're producing ATP, so you're taking the chemical energy that you're starting off with, and you're producing energy for the cell to function. Okay, so we're going to talk about these in great detail. You're going to have to know the basic steps of these. Remember, you're not going to need to know these in great detail. You'll see these in upper level courses. So, metabolism players. We're going to talk about cellular respiration, okay, and we're going to talk about photosynthesis. So, what we have are electron carriers, and there are two of them in cellular respiration. And the first one is nicotinamide adenine dinucleotide, which is NAD+. NAD+ will produce NADH, okay, because it is going to be reduced. So NAD+ goes to NADH when you add an electron reduction, right? Flavine adenine dinucleotide, FAD, will go to FADH2 because you're going to add an electron to it, and it's, again, going to be reduced. And we're going to talk about the way that the molecules deliver the electrons to the electron transport chain. So in cellular respiration, these two guys right here, NADH and FADH2, are going to go to the electron transport chain, and they're going to exchange their electrons for ATP. Just like you go to a currency exchange in a foreign country, you take your American dollar and they give you a Great British Pound, for example. Okay. In photosynthesis, the NADP+ is going to be reduced to NADPH. What does that mean? Reduced. You are adding an electron to it. So then NADPH is going to be carrying the electron. And we're going to talk about where NADPH is going to carry the electrons in photosynthesis. In both processes, you're going to see similarities, okay? First, both processes, you're going to see the process of chemiosmosis, which is basically you're going to have electron transport chains. And then in both processes, you're going to have ADP turned into ATP. So again, the ATP, why are we producing ATP? Why are we making ATP? What is all the fuss about ATP? Well, ATP is the chief energy currency of the cell. So you are going to use that ATP to perform functions in the cell. Very, very, very important. Okay, so the first process I want to talk about is aerobic cellular respiration. Okay, aerobic cellular respiration. We're going to break that down. Aerobic means with oxygen, and then cellular respiration is basically what you are seeing, the equation or the reaction. It's using the oxygen to convert it to carbon dioxide. Okay, respiration, just like you know respiration, you breathe in oxygen on a organ level, you as a human being, and then you exhale carbon dioxide. We're talking about this molecular, so it's cellular respiration. It's at a cellular, and we're using molecules, so it's at a molecular level. The reaction. This is a really good equation to memorize. You should just memorize it. For those of you going on to upper level biology courses, you will be seeing this concept over and over and over again. So it's just a good idea to memorize the equation now. But we're going to break it down and talk about every single player here in this general equation. So you start off with a molecule of glucose, which is C6H12O6. You add oxygen, so 6 molecules of oxygen. You're going to produce 6 molecules of carbon dioxide, 6 molecules of water, and you're going to produce 38 ATP. And we're going to talk about where those ATP molecules come from. Okay, the overall goal of aerobic cellular respiration: 1, you're going to break the bonds in the carbohydrates to release the electrons. 2, you're going to use the electron carriers, okay, to harvest the electrons so that you can carry the electron to the electron transport chain. 3, you're going to want to create a concentration gradient of electrons and hydrogen atoms that are going to want to cross the membrane. Because remember, when you have a high concentration, the molecules want to go to a low concentration. So it's always going back to the law of diffusion. And then what we're doing is finally, we're using that concentration gradient, okay, to fuel the ATP synthase, which is the enzyme, which is the transmembrane protein, which is AKA the integral protein that will take the ADP and the phosphate, bring them together to produce the molecule ATP. This is chemiosmosis. This is chemiosmosis, okay? Now, the process of aerobic cellular respiration occurs in 2 locations. First, you're starting off in the cytoplasm. So glycolysis will occur in the cytoplasm, and we'll talk later on why it occurs in the cytoplasm. And then the rest of the processes, which include pyruvate oxidation, the Krebs cycle, and the electron transport chain, all will occur in the mitochondria. Okay, so here we are. There are 4 stages. I like to call them stages, not steps, because each of the steps is a, again, basically the step of the reaction. So I'm just going to come in here while you guys are on, and I'm going to change it to stages, okay? Stages. And the 4 stages are glycolysis, the intermediate stage, which is pyruvate oxidation, the Krebs cycle, which is also known as the tricarboxylic acid cycle or the citric acid cycle, and then finally, the electron transport chain. So you start off with a molecule of glucose. It's actually, so which is 1 molecule of glucose, which is 6 carbons. You're going to go at the end of glycolysis, you're going to have 2 molecules of pyruvate, which is a sugar, but it's a 3-carbon sugar. So 2 times 3 is 6. And 6, remember, we cannot violate the laws of chemistry or physics ever, ever, ever. So we have to keep track of all our elements. Remember, they can change organization, but you cannot change the number of atoms of the reaction, okay? Then the 2 pyruvate molecules are going to be oxidized into acetyl-CoA, and then acetyl-CoA will go into the Krebs cycle, and then what you have is the production of electrons from these processes. So all of these processes, whether it's a 10-step glycolysis, the 9-step Krebs cycle, okay, you're going to have electrons which are being carried by the electron carriers, and then you're going to deliver them to the electron transport chain, and basically what's going to occur is you're going to produce the ATP. And the number again is 38 ATP. All right, so we're going to go over the steps of cellular respiration, okay? So I like to give overview of the entire process so that when we finish, you can come back to this diagram, which looks like a lot right now, okay? But we're going to break it down and we're going to go through it individually, and we're going to talk about all the processes, each stage: glycolysis, pyruvate oxidation, citric acid cycle, and then finally the oxidative phosphorylation. And our goal is going to be producing the ATP, okay? So without further ado, we are going to start off with stage one. Stage one is known as glycolysis. What you are doing in the process of glycolysis is you are taking one molecule of glucose, which is 6 carbons. You can count the 6 carbons, and you are producing 2 pyruvate molecules, which are 3 carbons each. Okay, you're using 2 ATP. You produce 4 ATP, but you've already used 2, so that means the net production is 2 ATP. Okay, so you're also producing 2 NADH, and with the process of glycolysis, look at that word here next to the bullet point, anaerobic. That means glycolysis will occur with or without oxygen. That means what? What can you take away from that statement? That glycolysis will occur with or without oxygen. Yes, all organisms on Earth will undergo the process of glycolysis. Okay, the reactants or the substrates are 2 ATP molecules and glucose, and the products are 2 pyruvate, 4 ATP, and 2 NADH. Okay, so look at this summary. The energy tally, okay, the amount of energy that we produced from glycolysis, a net gain of 2 ATP, so you should know how that occurred, okay? We produced 4, but we needed 2 for the first step, so 4 minus 2 is the 2 ATP. Then we produced the 2 NADH. What are we going to do with those? We're going to use those to continue the process of cellular respiration, okay? Stage 2. Stage 2 is pyruvate oxidation, okay? What you're doing is you're taking pyruvate and you're adding acetyl-CoA, and what you with a enzyme known as pyruvate dehydrogenase, what you're doing is you're breaking the pyruvate molecule down into from a 3-carbon pyruvate molecule to a 2-carbon acetyl-CoA. But what happens to the other carbon? Because remember, the reactants and the products, you have to have the same number of elements. Okay, so where does the other carbon go? It's released as carbon. Excuse me. Released as carbon dioxide. Remember, we produce 2 pyruvate molecules that in glycolysis. That means 2 pyruvates are being oxidized. That means you have two intermediate reactions. You're going to produce two NADH. Why? Because each reaction produces an NADH. So this is overall two NADH. And it is aerobic because the oxygen is going to accept the dispensed carbon. That's how you produce the carbon dioxide. Okay. So again, substrates, you're starting off in the intermediate stage or the pyruvate oxidation stage. You start off with two pyruvates, you have two oxygens, and then you have two CoA molecules. And the CoA is a coenzyme A. Again, remember we talked about coenzymes. They help to facilitate reactions. Okay, they help enzymes. So the products are going to be two acetyl-CoA, two carbon dioxide, and two NADH. And we're going to talk about what happens to the carbon dioxide. Everyone should know what's going to happen with the NADH. Remember, NADH is an electron carrier. That means it's going to transport the electrons to the electron transport chain. Yes. What about the carbon dioxide? Carbon dioxide is just going to diffuse out of the cell. Don't we produce carbon dioxide? Don't we exhale carbon dioxide? Yep. So let's talk about so far what we produced in terms of energy. 2 ATP and 4 NADH at the end of pyruvate oxidation. Now that's not a lot, right? That's not a lot of energy yet. We're still getting there though, okay? Now we go to stage 3 of aerobic cellular respiration. Stage 3, basically as I stated, the remaining carbons are released, okay? So they're gone from before. So what you're doing is the release of carbons is going to generate a lot of energy from the Krebs cycle. Now you don't have to memorize each of the steps of the Krebs cycle or of glycolysis. You'll do that when you get to biochemistry. What we want you to know here in 181 is what are the products that are being produced and how are we going to generate the ATP, that magic number 38, okay? So the acetyl-CoA, remember, is entering. Remember that there are two acetyl-CoA. So this is for each acetyl-CoA molecule, okay? Acetyl-CoA is going to go in through a series of reactions, okay? And it's a cyclic, that's why tricarboxylic acid cycle. It's a cycle. It's a citric acid cycle, or it's the Krebs cycle named after Dr. Krebs, all right? So you're releasing the carbons and you're generating energy in the form of what? Electron carriers, 3 NADH, 1 FADH2, 1 ATP. And that is per acetyl-CoA. How many acetyl-CoA molecules are going into the Krebs cycle? 2, because you had 2 acetyl-CoA per 1 glucose molecule. All right, now, do a review again. What are substrates? 2 acetyl-CoA molecules. What are your products at the end of the Krebs cycle? 4 carbon dioxide, 6 NADH, 2 FADH2, and 2 ATPs. So if we look at our energy tally up here in the box, what have we produced so far? We have 4 ATP molecules, we have 10 NADH molecules, and then we have 2 FADH2 molecules. What do we want to do with the NADH and FADH2? Remind us again. Correct. You are going to exchange those electrons for ATP. All right. How do we do that? Via the amazing electron transport chain using the process of oxidative phosphorylation via chemiosmosis. Yes, I know that is a lot. So we're going to be using the electron transport chain, which is going to allow for the process of oxidative phosphorylation via the process of chemiosmosis. So what are we doing with the electron transport chain? We're going to take all the energy that's in the form of electrons and hydrogen ions with the NADH up here and NADH here and NADH here and FADH2, all that we produced, okay, so far to generate ATP. So the electron pump, okay, the electrons are going to pump the hydrogen across the membrane because you're going to create a gradient, okay? You're going to create a gradient. So you are right here, excuse me. So we are right here, the production of the gradient. So NADH is going to approach what we call complex 1. FADH2 is going to approach complex 2. And then basically you're going to take those electrons and hydrogens off, okay? And you're going to build up these protons or hydrogen ions. And then you're going to create, what are we building here? The proton motive force that's going to go through, who's this guy? ATP synthase, okay? So remember, one hydrogen ion is going to produce one ATP molecule. How? ATP synthase is going to take ADP and the phosphates that are sitting in the mitochondrial matrix and smash them together to produce ATP. Now, let's talk about this. The NADH, which has 3 electrons, is going to produce 3 ATP. So 3 electrons in the form of hydrogen ions, 3 hydrogen ions equals 3 ATP molecules. Whoops, excuse me. FADH2 will only give off 2 electrons or 2 hydrogen ions. Therefore, you'll produce 2 ATP molecules. The electrons then will combine with hydrogen and oxygen to form a water molecule. 2 hydrogens and 1 oxygen, okay? So if we have a hydrogen and we have oxygen molecules and an electron, what are we doing? We're producing water, H2O, okay? So again, 1 NADH. So this is just a slide showing you how it's going to occur. So NADH approaches the complex, okay? And it's going to strip the electrons off of NADH. So it's going to strip 1, 2, 3 electrons, okay? And then you have the 1, 2, 3 hydrogen ions because the electrons, you're not actually stripping electrons, you're stripping hydrogen ions. So then hydrogen ions are then 1, 2, 3 from NADH going to produce 3 ATP. How? The hydrogen is going to go through ATP synthase. ATP synthase is going to take ADP and the phosphate, smash them together, and produce ATP. It's not really smashing together. You know what I mean, okay? Again, the FADH2, summary, remember it's 2. So FADH2 can only give off 2 electrons, so 1, 2 hydrogen ions go through ATP synthase, again, to ATP. Remember the fact that we are undergoing the chemiosmosis force, which means that we need to take those hydrogen ions that we produce and cross the membrane. Remember that hydrogen ions are too big and polar to cross the membrane. So who's going to help the hydrogen ions cross the membrane? That is right, ATP synthase. Yes, ladies and gentlemen, ATP synthase, okay? So the summary of cellular respiration. The 4 stages, you have glycolysis, the intermediate stage, also known as pyruvate oxidation, citric acid cycle or the Krebs cycle, and then the electron transport chain. So you can just go over that summary slide, and basically what you're talking about for each stage, you have the production of different molecules. So you start off with reactants, aka substrates, and you end up with products. Okay, glycolysis occurs with or without oxygen, so that means it occurs in all organisms. Okay, second, the intermediate stage, remember pyruvate that's produced, but you're producing 2, so it's double of everything after this. Okay, you're producing the acetyl-CoA and the carbon dioxide, and the carbon dioxide is just going to be waste. Okay, and third, the citric acid cycle. Remember, you're producing those electron carriers. I mean, you're not producing electron, you're producing high energy electrons, and electron carriers are going to be there to accept them. Okay, and you're producing a little bit of ATP. And then finally, the electron transport chain, we are taking everything and we are exchanging it for ATP. It's like going to, let's say, Europe with the dollar, and you're going to exchange it for the euro, okay? Because you can't use the places that you're going to go to are not going to accept the dollar, okay? It's the same thing here. We cannot use electrons. Our cells use ATP. So we need the ATP to perform cellular functions. So in the end, the 2 ATP from glycolysis plus the 2 ATP from the Krebs cycles plus a whopping 34 ATP from the electron transport chain will give you a total of 38 ATP in all for a single molecule of glucose. Yes, single molecule of glucose will result in the synthesis of 38 ATP. All right, so just go through the review again, different images, because I know I have a lot of different learners out there, a lot of you that like seeing images and diagrams to understand, you're visual learners, okay? Some of you like hearing my voice and your auditory when I'm explaining stuff, but basically it's the exact same stuff. Glucose, through a process of glycolysis, becomes pyruvate. Pyruvate will enter the mitochondria. You have the pyruvate oxidation, where pyruvate is going to be broken down into acetyl-CoA, and then acetyl-CoA goes into the Krebs cycle, where you produce those high energy electrons, which are going to be carried to the electron transport chain. And then the oxidative phosphorylation is going to produce the 38 ATP. All right. So again, another summary. Here we go. Without images, if you like seeing the words, which I know a lot of you do, okay, glycolysis, the 6 carbon molecule glucose will go with the 2 ATP that you start off with, will produce 2 3-carbon pyruvate molecules, 2 ATP, and 2 NADH. The intermediate or pyruvate oxidation stage, you take 2 3-carbon pyruvates and you convert them to 2 acetyl-CoA molecules plus 2 carbon dioxide plus 2 NADH. The Krebs cycle, the 2-acetyl-CoA produced from the pyruvate oxidation will go in to the Krebs cycle. You'll produce 2 carbon dioxide, 2 ATP molecules, 6 NADH, and 2 FADH2s. And then finally, you're going to take the NADH and the FADH2 generated via glycolysis, pyruvate oxidation, and the Krebs cycle. And with the 10 NADH, you're going to produce 30 ATP. Remember, 1 molecule of NADH produces 3 ATP, so 10 times 3 is 30 ATP, and then 2 molecules of FADH2, 4 ATP, because 1 molecule of FADH2 produces 2 ATP. So 34 ATP from the electron transport chain, plus 2 ATP from glycolysis, plus 2 ATP molecules from the Krebs cycle, generate the total of 38 molecules of ATP. 38 molecules of ATP. Now, to give you an idea of how much ATP you use, okay, the average cell will utilize or use, not produce, use, the average cell will use 2 million molecules of ATP per second. Yes, you heard me right. A cell will utilize 2 million molecules of ATP per second. Yes, that's huge. Now think about this. You, as a human being, on average, okay, during a normal 24-hour period without exerting yourself, okay, will utilize 90 pounds of ATP per day. Yes, 90 pounds of ATP per day. That's on average. Now, if you add an exercise or whatever, it's going to increase. But just think about that. 90 pounds, for those of you that go to the gym, that's 2 of the 45-pound plates at the gym. So imagine when you carry those weights in your hand, that's how much ATP you're using in a single day. 90 pounds. That's extraordinary. That's extraordinary, isn't it? Okay, so that's why ATP is so important. Okay, so you can see again, just a table showing you the energy totals. If you didn't like the way we had it here, we have it in a table format here for you. So you can see again, what do you produce in glycolysis? What do you produce in intermediate stage? What do you produce in the citric acid cycle? You got a total of 38 ATP. Okay, again, what are we doing in metabolism? You can take all of the foods that you consume. Well, everybody knows about carbohydrates. So we're taking the carbohydrates, complex carbohydrates like starch, we break down into glucose, and then we go through that process. But did you know that when you consume fats, you can also push it into the pathway to break down? It can be converted to sugar. And you can also take amino acids from proteins to produce the ATP. So all of the macromolecules that we talked about, 3 out of 4, even all 4 of them can be converted to ATP. The bottom line, you want to produce ATP. All right, so you can see all of the ATP that we produce are going to drive the synthesis reactions. So all the synthesis reactions that occur in an organism, we need ATP for it. Okay, respiration, breathing. Okay, so again, on the the main level, for example, the organism level, take in oxygen, okay? And you're going to take that oxygen, go through the lungs, deliver it to the tissues, deliver it to the cells. The cells will undergo cellular respiration, and then the carbon dioxide diffuses out of the cell, out of the mitochondria first, out of the cell, into the bloodstream, to the lungs, and then finally into the atmosphere. So it's a cycle. Take oxygen in, get rid of carbon dioxide. All right. Aerobic versus anaerobic. So aerobic cellular respiration, okay, you're going to go through a process that we talked about and produce 38 ATP. With anaerobic cellular respiration, okay, we are not going to have those processes occur because we don't have oxygen. So what happens is you can have fermentation occur. And with fermentation, you have an organic molecule that is going to be accepting the electrons. Because the goal is to regenerate NAD+ so that glycolysis can continue. So I think I'm going to stop here for now, and I will pick up on our discussion of fermentation and cellular respiration without oxygen. Actually, no, I'm going to wrap it up because it's just one more slide. So basically, sorry about that. Basically, with fermentation, we need the NAD+ for glycolysis. Remember that during the process of glycolysis, we're going to produce electrons, so we have to have NAD+ there to pick up the electrons. NADH is generated via glycolysis, remember, because you're reducing the NAD+ to produce NADH. In fermentation, there's no electron transport chain because glycolysis does not use electron transport chain, so NAD+ is not regenerated. NAD+ becomes depleted, NADH accumulates, and that will lead to eventually halt energy production. You do not want that to happen in a cell or in you as an organism. So what occurs is fermentation is going to use an organic molecule other than it's going to use another organic molecule as an electron acceptor in order to regenerate the NAD+. So you go through glycolysis, you produce your two pyruvate molecules, but instead of the pyruvate molecules going to the mitochondria and pyruvate oxidation occurring, the pyruvate molecules are going to be converted into something else. And depending on what kind of organism you are, okay, if you're an animal, pyruvate will be converted to lactic acid in order to regenerate NAD+. And if you're another organism, for example, like fungi or yeast, some bacteria, you will produce ethanol. Okay, so again, very, very important. And this does occur. Obviously, we don't want it to happen in us. So let me give you an example of when we undergo fermentation. Let's say you're working out really hard, and you all of a sudden get a cramp. Well, how did that cramp arise? Where'd you get it from? Why are you feeling that cramp? Well, basically, what happened is you were driving and pushing your muscles so hard that oxygen could not get there in time. So it wanted glycolysis to continue. So what happens is it went and it oxidized the pyruvate molecule. The pyruvate molecule was reduced to lactic acid in you. And lactic acid actually burns muscles. And that's why you get that crampy feeling. It's a muscle burn, okay? Another way, fermentation in other organisms, for example, yeast, okay? We will drive yeast, okay, in low oxygen environments to produce drinking alcohol. And that's how we make beers, wines, so on and so forth, okay? So you can see the summary: 3, aerobic respiration, which we talked about in detail, anaerobic respiration, and fermentation. I do want to talk about some another anaerobic respiration process. So, and it was in the reading, and it was one of the questions. Basically, when we start exercising as human beings, the initial exercise period, or the period where we are not getting oxygen to the tissues. Now, what does that mean? I mean, it doesn't mean that we cut oxygen off completely. If you cut oxygen off completely for a certain amount of time, for example, 4 minutes to the brain, you're going to die. What happens is you don't have oxygen arriving there yet. It hasn't gotten there yet, okay? So you have a molecule called creatine phosphate, which will generate ATP for about 2 seconds for each molecule. And you have a lot of creatine in your muscles, and you have a lot of creatine phosphate produced. So creatine phosphate will undergo a hydrolysis reaction. So basically, it's substrate level phosphorylation, taking the creatine and the phosphate molecule and producing ATP. So the ADP plus the phosphate from the creatine phosphate will produce ATP. So I just wanted to mention that. Okay. So basically go over the process of cellular respiration. Most of it is aerobic cellular respiration, but do study the anaerobic respiration, what happens to pyruvate when oxygen is not present. So you should read that too in the book. So I'm emphasizing that. So what happens to pyruvate when oxygen is not present? Those are the questions you have to answer. All right. So that is the end of chapter 6. And I will be back to talk to you about photosynthesis. All right. Take care, everybody.